So hi everybody, welcome to the HIRM seminar for today. It's a great pleasure to have Dr. Chris Burge with us today. Chris is a professor in the biology department at Massachusetts Institute of Technology in Cambridge, Massachusetts. He performed his bachelor's at Stanford University in biological sciences, and then received his PhD also from Stanford in computational biology before working as a postdoc in Phil Sharp's lab at MIT in computational biology. And since then, he has moved up the ranks at MIT from a fellow to an assistant professor, associate professor, and now full professor. It's really wonderful to have Chris with us today. He's an expert in RNA metabolism and most recently in protein-RNA interactions. And today he'll tell us about protein-RNA interactions in vitro, predict in vivo binding and regulation. Welcome, Chris. Thanks for coming.
Thank you. Thank you, Frank, for the invitation. It's great to be here. Thank you, Jay, for organizing as well. So today I want to tell you a bit about our work starting from in vitro characterization of protein-RNA interactions, and then asking how the lessons we learn in vitro can be extended to predict in vivo binding and regulation. And I'm used to lecturing by Zoom and having people chat questions and so forth. So please feel free to chat questions during the seminar, and I will either answer them directly or Frank will ask them. Thank you. All right. So RNA binding proteins, or RBPs, here regulate many aspects of gene expression, both prokaryotes and eukaryotes. My lab is interested in RBPs predominantly in mammalian systems. We've recently been working on human RBPs with the ENCODE project. And of course, they regulate not only RNA splicing, which is a sort of a core activity in the lab, but also cytoplasmic phenomena such as mRNA stability, localization, translation, and so forth. Many RBPs are essential for cell viability, making them somewhat difficult to study by knockout, but they can frequently be depleted to study their functions. And importantly, the presence of all these RBPs and RNA interacting with RNA structures offers diverse opportunities for therapeutic intervention. Okay, so today I want to tell you a little bit about our multi-omic analysis of human RBPs. So this was started as a ENCODE phase 3 project with several collaborators, and is continuing as sort of a genome resource project. And we want to tell you about the data and also how to access it, and we hope that it's useful in your research. And then in the second half, I want to tell a little bit about our work on in vitro measurements of protein-RNA interactions and how we're using those data to then predict aspects of their in vivo functions. So several years ago, I got together with our friends, Brent Gravely, Jean Go, Jiandong Fu, and Eric Lecuyer, and worked on a project to characterize human RBPs at a large scale. And so the assays that were used included my lab's RNA binding seq for in vitro protein-RNA interactions, which I'll tell you about more in a little bit. The Yo lab developed their eClip assay, an enhanced cross-linking IP assay to measure protein-RNA interactions in vivo. The Gravely lab studied RBP function by knocking down RBPs with short hairpins and doing RNA-seq analysis, from which you can see changes in mRNA abundance or splicing, of course. The Fu lab studied some of the RBPs that are nuclear in localization by chip assay to ask about their interactions with chromatin. And Eric Lecuyer's lab in Montreal studied the subcellular localization of RBPs using immunofluorescence and, in some cases, GFP tagging approaches. And so our goal, sort of long-term goal, is to make a sort of a periodic table of RBPs to sort of help organize RBPs into groups of similar structures and functions so that you can kind of look up any RBP and know where it's expressed in the cell, what its binding motif looks like, what its distribution of binding sites across the transcriptome looks like, and some idea of what its function may be. To date, or as of our publication last summer describing sort of the initial effort in this direction, we've characterized over 300 human RBPs from diverse structural and functional groups. Many of them have RRM domains, the most common type of interaction domain, but others have zinc fingers, KH domains, or other types of RNA binding domains. Our initial characterization was perhaps a bit biased toward more well-known factors. We want to include sort of some positive controls, but also includes many uncharacterized RBPs as well. And it includes proteins with functions in RNA processing, export, stability, translation, many other functions. So one analysis that proved quite interesting was to look at where the clip peaks land in the in the transcripts. This very simple analysis of just grouping the RBPs by the fraction of clip peaks in either coding regions, UTRs, introns, and so forth, already can highlight cytoplasmic versus nuclear proteins, proteins involved in splicing, proteins involved in non-coding RNA processing, and so forth. And just kind of one of the examples of the types of things that you can do, when you have systematic data of the same quality across hundreds of different RBPs. Another thing that was of particular interest for those of us who work on RNA splicing was to sort of systematically develop RNA maps for each protein. So an RNA map in splicing is originally described by Bob Darnell, is a pattern of location-specific activity of the RBP. And so traditionally, one knocks down the factor and then looks for exons whose splicing changes and finds exons that are excluded upon knockdown, therefore likely activated by the protein, or included upon knockdown, therefore likely repressed by the factor, and then compare the sort of make a meta analysis of the clip peak density along the transcript for the upregulated and downregulated exons. And what you can see here at the top for RBFOX2 protein is this very prominent peak here downstream of the exon in exons that are excluded upon knockdown, indicating that RBFOX activates splicing when it binds in this location. And similarly for the below, for PTBP1, we see a prominent red peak upstream and in the exon, suggesting that PTBP1, as is well known, represses splicing when it binds either in or near the 3' splice site or in exon. And so these can now be sort of automatically generated for all the RBPs for which we have, which influence splicing, for which we have knockdown and RNA-seq, and clip data. So here's an example of RNA maps for all of the HNRNP and SR proteins that were analyzed in this study. Here, as part of the ENCODE project, we analyzed primarily K562 cells and erythroleukemia line and hepatitis 2 cells, a liver cancer line. So that's the purple and green for each of these. And then there were biological replicates of the clip experiments that are shown as well. And so you can see that this very prominent pattern where the HNRNP proteins predominantly repress splicing when they bind within the exon or nearby, whereas SR proteins predominantly activate splicing when they bind within exons. And then there are other patterns that sort of subtle differences between different proteins as well. And I want to emphasize that this particular analysis was not a surprise. It's been known that SR proteins and HRMPs have these general activities, but this kind of demonstrates the sort of the way that this can be done in a large scale, and then we can make RNA maps for many other factors, including those that are less studied. Okay, so today I mostly want to focus on our in vitro studies, and so we have been performing this assay we call RNA binding seq, or RBNS, where we clone either the full-length human RBP, or sometimes if it's too large, we just clone a region containing all of the RNA binding domains that are known in that factor. We express a tagged version in E. coli, purify it, and one of the tags that we use is a streptavidin binding peptide tag, which we can then use to immobilize the protein on beads, and then incubate with a pool of random RNA, which we make by in vitro transcription. So typically, we use a random 20 mers or random 40 mers with sort of minimal adapter primers on either side. And we use random sequences in order to give us the greatest diversity. So it's a sort of unbiased assessment of what the protein will interact with. We've also, in some cases, used natural sequences, which we make by array synthesis, for example, to study here. But for this sort of large scale analysis, we wanted to use random sequences. And I want to mention that my lab, this sort of foray into biochemistry was partly in order to create sort of a gold standard for CLIP. So when you're doing CLIP at a large scale, if you have a well-characterized protein, then you see, it's RB-Fox, and you see the GCAUG enriched in the CLIP peaks, and your CLIP is on track. But if it's some unknown protein where there's nothing known in vitro, it's very hard to know whether the CLIP is working and what the possible false positive, false negative rates of CLIP may be. So we wanted to have a sort of orthogonal standard here. Okay, so with the protein immobilized, we add RNA. We typically do this assay at multiple different protein concentrations, ranging from low nanomolar to low micromolar. And we use a concentration, sort of total concentration of RNA of around of 1 micromolar typically. So then we isolate and we elute the bound RNAs and subject them to deep sequencing. And then we compare the enrichment of short polynucleotide motifs in the bound pool relative to a control input pool of RNA, or sometimes we do a no protein sort of matched control. And so, for example, for this protein H&RBL, here we are looking at sort of pull down reads versus input reads. And if you look at this particular, we typically look at 5 mers, 6 mers, 7 mers. So for example, the 5 mer ACACA is roughly 4.5-fold enriched in the pull down reads relative to the input reads. So we describe that as the R value, or raw enrichment value of that factor, so defined as the rate by the ratio of the frequencies in pull down to input. And so we developed a pipeline to sort of automatically calculate the R values and then align the enriched k-mers into motifs, which yields the motif shown here, ACACA, with some possible U substitutions. And we've done that now for, actually at the time of publication, we've done 78 human RBPs, and now we've gone on to here over 100. Although I'll mostly talk about these 78 today. And so here are the data, including the domain structures, as well as the motifs for each of the 78 RBPs that we characterized. And in this analysis, we cluster them not by their phylogenetic relationship, as is often done, but instead by the relationship between their motifs. And so what you'll see here, it's actually quite striking, is that there are many different RBPs that bind poly-U, okay, shown here. There might be subtle differences, but all these proteins more or less bind poly-U, and that includes proteins that have different domain structures. So both RRM proteins and KH domain proteins bind poly-U, suggesting that they independently acquired that particular specificity. And similarly for poly-G, for various AU-rich sequences, for GU-rich sequences, AC-rich sequences, and so forth. And we analyzed the motifs fairly extensively and found that it is likely not a random subset of motifs in any sense. There's a pronounced tendency for clustering, even if you eliminate any close homologs within the set. Also, the sequences are predominantly low complexity sequences. Not all of them. For example, RB Fox binds GC-AUG, which is not low complexity, right? But many, but low complexity sequences are much more likely to represent the motifs for RBPs than others. And they have other observations, and we're still sort of puzzling through this and trying to understand the overall organization of the specificities of human RBPs that we're happy to talk about. Okay, so when we compared the motifs obtained in vitro to motifs that we obtained by running the same pipeline on the clip peaks rather than the pull down reads, we often got very similar motifs, as shown here for FUS, finds a G-rich motif, HNRPK, a C-rich motif, HNRPL, AC repeats, and so forth, either measured by overlap between the most enriched pentamers or by sort of various correlation measures shown here. So in most cases, about 80%, you get more or less the same motif, but then there are some exceptions where different motifs emerge as well, and we're looking into this. Okay, so one of the things that we noticed was that, of course, some clip peaks will contain a high affinity motif, and others will not. And so we asked, do the clip peaks that contain a motif have any differences in terms of their associated regulatory capacity? And what we found is that actually you see stronger delta signs, that is, stronger changes in the percent spliced in of regulated exons when you look at the subset of clip peaks that have the high affinity motifs versus other clip peaks for the same factor that lack these high affinity motifs. I'm suggesting that that perhaps those with high affinity motifs represent locations of longer duration, more stable binding that may be required for regulation. So we think that the RBNS motifs may be useful in sort of categorizing different clip peaks in terms of their reliability. All right, and so could we do more than just develop a motif for each RBP from these data? So we have data for 78 proteins at 5 different protein concentrations. Could we leverage that to infer biophysical properties of the binding? And so that's when postdoc Marvin Jens joined my lab a couple of years ago, and he had a biophysics background. He was training in Berlin and wanted to develop a sort of a proper biophysical model to analyze these data. And the model he developed is called RBP-AMP. And I just want to tell you sort of a few features of this model. So it begins by looking for enrichment of K-mers as before. In his case, he starts by calculating the enrichment of all 8 mers. So here for this protein, MSI1 or Musashi1, it likes to bind UUAG with very high enrichment. He then clusters these 8 mers to form alignments. So similar to the previous pipeline, to create an initial set of what he calls position-specific affinity matrices. And so one of the differences here is that we allow each protein to have multiple different matrices, up to 5, just sort of for practical reasons to describe their affinity. We found that there's some proteins that can be described by a single matrix, such as RB Fox, but others seem to require multiple distinct matrices to accurately describe their their specificity. So these PSAMs, or position-specific affinity matrices, are matrices in which each column is a different position, and there's a 1 at the corresponding to the base that is the sort of the highest affinity optimal base. So the canonical motif with a perfect consensus at each position would have the product would be 1, and then there's sort of a scaling factor, which is the KD of that optimal motif. And then all the other entries in the PSAM are sort of, say, 0.5, 0.1 would represent a 2-fold loss in affinity relative to the optimal motif, a 10-fold loss, and so forth. And so you just multiply the entries in the PSAM to get sort of the relative affinity, and then you scale them by the KD, which we estimate independently. Okay, so we make this initial PSAM set from these 8-mer alignments. Then we do a stochastic gradient descent, which I'll talk about more in a moment, to get optimized set of PSAMs. Then we add in RNA secondary structures. So of course the RNAs in solution are folding, and so we model that with standard thermodynamic folding algorithms, and the folding is competing with the binding. And so we kind of have a separate analysis that asks how much does the protein care about whether its motif is folded or not. Most of our proteins strongly prefer to bind single-stranded RNA, and do a second round of the stochastic gradient descent to the final PSAM set. So here's an example for this Musashi protein. If we require it just to bind to a single PSAM, we get a UAG containing motif. But if we allow it to have multiple PSAMs, we get a set of motifs shown here, which have UAGs with variable spacing, suggesting interaction between multiple RNA binding domains with a little bit of flex in between. And then we can actually estimate a KD for each of these motifs as shown. Okay. And this is a pattern that we observe for many RBPs, that we get. We can better describe the data with more motifs, not perhaps not a surprise, but that the motifs often are related to each other by the presence of 2 short submotifs with variable spacing. You see that here for Nova. You see it here a little bit for TRA2A, for example, for HNRP0, and others. Now, in other cases, we see other patterns. So this is kind of an interesting one. This protein, also known as roquin, we see a core UAU motif. But then if you look at the different PSAMs shown here, you see that the flanking sequences, the flanking 3 bases on either side of the UAU are very often complementary to each other, suggesting that it binds that UAU in a stem loop conformation, which is actually consistent with literature on sort of biochemical studies of this roquin protein. Okay. So RBP AMP includes a number of features that have been sort of often ignored or glossed over in other types of models. So we, actually there's a question here from Fabrice Leclerc, are the more conserved residues in motifs also those that contribute more to binding or to binding specificity? The answer is generally yes. We've only looked at that for a subset of proteins. We haven't yet done that on a systematic basis. But initial answer, yes. Okay. So the model, instead of using a linear approximation, which is often used, assuming that the protein will be dilute in solution, we actually model saturation because we're using the protein at both from low nanomolar to all the way up to micromolar, which is fairly high for a protein that has a KD of low nanomolar, for example. And so the optimal motifs are going to become saturated. And so we actually put that into the model, although it makes it more complex. Also, we can't assume that the free protein concentration is equal to the total concentration because we have a significant amount of RNA there as well. And so some protein, a significant amount of protein may be titrated by the RNA. And so we actually model the free as different from the total. And then, as I mentioned before, we also model the competition between protein binding and RNA folding. Another question about from Kafer Ostemir about, are you using the thermodynamic minimum folding free energy or co-transcriptional folding? Yeah, so this is, we are using the sort of standard MFE from RNA fold. These RNAs are kind of in solution for a considerable amount of time. They're simply going to equilibrium, and they're very short also. Okay. All right, so here's an example. So for example, for the TRA2A protein, the function that we've chosen to optimize is the square difference between the predicted 6-mer enrichments and the observed 6-mer enrichments. And so that's kind of what's shown here. Observed on x-axis, predicted on the y-axis, and I'm showing 2 different concentrations here of the protein. And you can see initially we get a correlation of either 0.9 or 0.84, so it's fairly reasonable from the initial assignment of PSAMs. But then after we do the stochastic gradient descent, we're able to sort of optimize the parameters being the entries in the PSAMs, as well as the KD of each PSAM, as well as a single quantity that describes the background in each experiment. And now we're able to get a quite good fit between predicted and observed 6-mer enrichments. And this just shows how this stochastic gradient descent looks in terms of model error. It declines initially, then flattens. This is when we do it without considering RNA secondary structure. Then we add the secondary structure. It is initially worse, but then it eventually achieves a new optimum. And we've achieved correlations on the 0.9 range are often better for most of the proteins that we've analyzed to date, as shown here. And it doesn't seem to be restricted to proteins with a particular domain structure, etc. We can generally do proteins with a correlation of 0.9 or better. There are some exceptions. So a couple of the proteins that I've highlighted in red here, such as this rope-win protein that I mentioned before, RC3H1, as well as ZNF326, where we do less well, they're known to bind secondary structures. And so our model actually assumes that the protein prefers single-stranded RNA. And so we know that that's sort of a limitation of the model. It's never going to get those proteins really perfectly. Okay. So because our model has several moving parts, we've actually systematically kind of pulled out different pieces of the model to ask how much worse does it get when we, for example, assume linear binding as a function of concentration instead of modeling saturation, and that actually has a big effect on the accuracy. And similarly, if we require a single PSAM instead of allowing up to 5 PSAMs, that also makes it significantly worse. And then RBP titration and RNA secondary structure have a more modest effect on accuracy. So just a little bit more on how we model secondary structure. So one thing we notice is if we take, for example, a well-characterized protein like RBFOX3, which binds, so this, in this analysis, we're looking at the 7-mer UGC-AUGU, which is sort of the optimal 7-mer for this protein. And then we then fold all the RNAs that contain, which is millions, maybe not millions that have that, but millions in total, perhaps tens or hundreds of thousands that have this particular 7-mer. And we calculate the accessibility of that 7-mer from each one. Then we can look at the enrichment of RBFOX on this 7-mer for the 7-mers grouped by different bins of accessibility, which is what's plotted here. And we can look at how that varies as a function of protein concentration. So what you can see here is that particularly at low protein concentration, there's almost no enrichment when the accessibility is very low. So when it's got a very strong fold, RBFOX does not bind. But then we see up to a 200-fold enrichment for very accessible incidences of this 7-mer, which then declines somewhat as protein concentration increases, and the protein can sort of more effectively compete with the with RNA secondary structure. And so one way to look at this is to look at the probability that for each base in a motif that it's unpaired, and that's shown here for data from each of the protein concentrations as well as input, and you can see, it's systematically higher. And looking at this distribution, as we expect, but looking at this distribution, it suggested that perhaps we could infer the region of the RNA that the protein interacts with and requires to be single strand from kind of looking at this pattern. And so we kind of did a fit there, and we're able to fit for RB Fox, which in this case is represented by an 11 base PSAM. We can infer that it's really the core 7 bases that the protein requires to be single stranded, and there's an offset. The optimal offset is 2 from the start of this motif, corresponding to the sort of UGC-A, UGY here. So we call these sort of RNA accessibility footprints, and in this case, the 7 bases corresponds quite well to structural data, but we have footprint for many other proteins and hope to then compare them to future structural data as those emerge. Okay. All right. So what about
Chris, Chris, you have some questions in the.
Yes.
You have some questions that you want to.
Oh, many questions. Do you want to read one of them of your choice, please?
Okay. For proteins that don't give you clear motifs, do you look at the predicted structure of the enriched oligos to see if they have a similar folding?
Yeah, great question. We have done that. It's not an easy problem. We often see essentially because in experiment we have the random region flanked by these 2 primary sequences that are fixed. If a protein really likes to bind, say, all stem loops or something, you may see a motif here in the random part that is complementary to the adjacent primer sequence, and that may show up. But it's tricky to tell, does it like to bind to that motif, or does it like to bind because it pairs with the primer? Of course, if we see enrichment of 2 different motifs, which bind to the different primers, then that would suggest that it's probably more likely to be structure. And then so what we do in that kind of situation is we then look for sequences that can fold internally to the random region and look for which structures are most enriched. And that works in some cases. So we have a couple cases that we highlighted in the Dominguez et al. Paper where we were able to infer structures. In one case, it was sort of like to bind a CA bulge out of a duplex, for example. But I don't think optimized that yet. So that's sort of an available project for an ambitious computational student, I would say, in the future to really, optimally tease that out from these data.
And then one more question: does your model distinguish between folding and unfolding equilibrium and binding equilibrium?
Yeah, I mean, it's kind of all modeled as one equilibrium system, essentially. And we don't handle kinetics. I should emphasize that we don't have a good way of measuring separately measuring on rates and off rates, for example. And we do the experiment. We incubate, for 1 to 2 hours, so presumably things are close to equilibrium. So thanks for the questions. Okay, so what about predicting in vivo binding? So here, what we've done is to basically take the RBP AMP motifs with their associated KDs and slide them along various transcripts. You could do this on pre-mRNAs. Here it's for mRNAs. Shown data here for 6 or 8 different proteins, and then the red shows the RBP AMP affinity, these kind of spikes down below, and then we're comparing that to clipped data. This is eClip data from Yo Lab, and I'm showing 2 different replicates here in different colors of blue or green, just to get a feeling for the variability in the clip density. And you can see that, for example, for the HNRPC case in the upper left, there are 2 prominent clip peaks, and those actually correspond to the 2 highest affinity sites in that gene. You can also see that the prediction that most of the binding occurs to the 3' UTR is also consistent with the CLIP data. Like here for L, it's more complicated. There's lots of different binding sites predicted, but again, there's kind of an overall correlation, certainly not a perfect correlation, and the magnitudes. I guess the locations seem to be better predicted than the magnitudes of the CLIP peaks would be one way to describe this relationship. But there is generally a reasonably good correlation, suggesting that first of all that the protein's binding specificities are similar in vitro and in vivo, and that the in vivo locations are driven to a significant extent by sort of the thermodynamic stability and don't require, for example, modeling of kinetics, or not radically redistributed by, for example, RNA secondary structures or RNA helicases pushing them around in this case. I'm sure that so there are cases where there's clear discrepancies, and those are likely to reflect the many differences with the in vivo situation where you have binding with with partner proteins, you have competition from other proteins or microRNAs, you have secondary structures, blocking accessibility, and so forth. And so here's one more example of TROP-PK binding to the coding region of this message. And so one of the things that Mark is interested in is to try to describe the in vivo binding in a more quantitative framework. And so the way he approached that, so he was thinking, about, well, it'd be great if we could sort of get a classical curve where we look at something that's proportional to occupancy as a function of the KD, and that would allow us to infer things like the the free protein concentration, or the whether there's cooperativity or not, and those sorts of things, which we can't really obviously we can't infer that in vitro, and the RNS assay is not good for measuring cooperativity. And so the way he did that was to essentially search the entire transcriptome in 50 base windows for regions of different relative affinity to each of our proteins. So for example, for the RB-FOX2 protein, you could slide along the transcriptome. In this case, this is 3' UTRs, and classify each 50 base pair window according to its relative KD. So here 1 represents the KD of the optimal motif for that PSAM, so a really great motif, probably UGC-AUG or something like that. And then obviously 10 would be 10-fold weaker than that, and 100 would be 100-fold weaker than that, and so forth. And then because the CLIP density is rather noisy, and we don't know that CLIP read counts are quantitatively reflect protein binding, what we do here is we take all of the 50 base windows across the transcriptome which have a high predictive affinity, and kind of throw them into one bin, and then look at the average CLIP density. So the idea being that the average may be more, should be more reliable than any given measurement. And then he looks at different bins and looks at the eCLIP enrichment shown here. And what we see here is that there's a nice monotonic decrease in CLIP enrichment as a function of KD, suggesting that there is, this is a good way to look at it and sort of confirming in another way that the predicted affinity does correspond to in vivo binding. And then he can sort of fit that curve and estimate parameters that may be related to. I want to estimate, like, we don't really think we can estimate these. We haven't shown that these are exactly what these parameters mean, but they're parameters that are analogous to free RBP concentration, and another parameter that reflects the slope that is analogous to a Hill coefficient or measure of flux. For most of the proteins, we fit a Hill coefficient of 1. That's suggesting lack of cooperativity, and that's consistent with the literature. But I'll show you some other counterexamples in a moment. Right. So this is just.
Hey, Chris, there's a question from Fabrice again. I think he's asking what many people are thinking. if you have cases where the RNA binding protein has multiple RNA binding domains, are you measuring some average binding, or do you know, which RRM you might be measuring?
Yes. So many of these proteins have, 2, 3, or more RNA binding domains, and we haven't systematically, mutated the different RNA binding domains and done the measurements to see exactly which one is binding, which motif disappears when you mutate this domain. But we believe that in many cases that we're using 3, 4, or 5 different PSAMs to represent that protein, and those may reflect the binding of different RNA binding domains. And each one, is assigned its own KD, and so often there's sort of with multi-RRM proteins, there's like one that has the really good high affinity binding, and then a couple others that bind more weakly, and so we are kind of capturing the aggregate binding we believe in many cases, and we're taking into account when he slides across these 50 base pair windows, he's sliding all of the PSAMs together. Well, so we're capturing the different binding modes that we can estimate from the in vitro data. Yeah, and so we typically estimate a free RBP concentration that's somewhat above the estimated KD of the optimal motif, as shown here. And a couple of interesting examples. So one protein that had where we estimate like which has a very unusual curve, which looks like it's saturating both the high and moderate affinity motifs, suggesting like a really, really high free protein concentration, this PCBP1 protein. We looked into the subcellular localization data from the Lecuyer lab, and found that it's very punctate in nuclei. In fact, it seems to co-localize with nuclear speckles, or sort of the site, the classical site where protein splicing factors accumulate. And so what we think may be going on here is that most of the clip may be coming from PCBP1 in these speckles, because that's where most of the protein is. And in those speckles, it's very highly concentrated, and therefore is saturating its high and moderate affinity binding sites. So there might be sort of two pools, another pool out in the cytoplasm, but it's harder to see where it's at lower concentration than would be binding to a different, when you would only be binding the highest affinity sites, for example. Another interesting case was the FUS protein, well studied because of its involvement in ALS. And in this case, we got another very strange curve here, where we see essentially no clip enrichment until you get to a KD, relative KD that's less than 1. So what does that mean? So again, we have a KD of 1 would be a single optimal motif within the 50 base window. Okay, but these motifs are typically about 8 or 10 bases long, and so one can achieve a KD or a relative KD less than 1 by having multiple optimal motifs within that window. And so only when you have multiple motifs in that window to get this relative KD of 0.3, 0.2, 0.1 do we see strong clip enrichment, and then it rises very, very steeply. And so this suggests that the free protein concentration is relatively low, but the protein binds in a highly cooperative way with an estimated Hill coefficient of around 5, which is unusual for RBPs, but is actually consistent with data from the Czech lab for FUS, where they did gel shift experiments and essentially saw like no binding, no shift, and then all of a sudden they see a huge shift consistent with several molecules of protein binding to RNA, and they can only see binding to RNAs of a certain minimum size, suggesting that like a single motif just is not sufficient for binding in FUS. It binds in a highly cooperative way. And so we'd like to know for the other proteins where cooperativity is predicted, what's going on there. And then finally, we wanted to ask about predicting regulatory activity. And so here, with these knockdown RNA-seq data, we we're interested in factors that might influence mRNA stability. So here, IGF-2, BP2 would be an example. And in this case, the way we analyze the data is a little bit different. We want to be able to compare CLIP data to RBP-AMP predicted affinities to other measures, other predictions of affinity. And so what we've done here is to ask each of these methods to rank the mRNAs by the predicted affinity. So CLIP will rank it by, CLIP read density relative to control, for example. RBP-AMP will rank the messages by predicted affinity. We can also use the, say, just the 6-mers or the best R values, or we can use other methods here. And so we have different rankings of the messages. And then what we look at is the fold change in mRNA abundance when we deplete IGF-2/BP2, or rather when the Greatley lab depleted IGF-2/BP2. And in this case, this is a destabilizing factor, so mRNAs tend to increase in abundance when you deplete IGF-2/BP2. And we can ask which of these methods best predicts the sort of the ordering of the sort of puts the messages that change a lot kind of at the high end of the rankings. And the way we do this is we calculate a cumulative log fold change z-score. We just add up the z-scores for each mRNA in order as predicted by each of these approaches. And so what we're looking for is, like what we see here for the CLIP enrichment, a very steep increase, indicating that the ones that change the most are the ones that are ranked highest by that protein. So here we get a nice excursion for the E-CLIP enrichment, and then a more modest relationship with the RBP-AMP affinity. It turns out that these excursions of these z-scores can be described by something called a Brownian bridge. Sort of has well-known statistical properties, or you can just simulate it, and you can estimate the significance fairly easily. And so, just for reference, these faint gray curves, those are like randomly permuted gene lists there. So you can just see that's kind of the background if you had a random ordering. Okay, so that's what we get. So CLIP clearly does an excellent job, and RBP-AMP not so great for this particular factor. But then for other factors, for example, KHSRP, we see that the RBP-AMP actually does a better job than CLIP at predicting which messages will change the most. And so here we've actually compared each of these different approaches. I won't talk about the orange one, but we'll focus here on the black, meaning CLIP, versus the red RBP-AMP. And we can see that in many proteins, the RBP-AMP does as well or better at predicting the ordering of regulatory targets than you can do with CLIP. And I just want to emphasize that CLIP is great, but CLIP applies to a particular cell type or tissue, and having sort of an a priori model of affinity, you can then apply that in principle to any cell or tissue, as long as you know what mRNAs are expressed there. So we do think this is going to be a useful tool for predicting regulatory targets in different cell types.
Chris, can I ask a question? Sorry,
sure.
But why do you think, I'm a little bit surprised, I guess, that RBP-AMP predicts it better than CLIP for a set of proteins. Why do you think that is, and what is different about those set of proteins that is better predicted this way than versus E-CLIP?
Yeah, that's a good question. There could be biological features, or there could be technical features that have to do with, how good that particular CLIP experiment is, or how good this particular RBS experiment is, or how good our modeling of it is. And so IGF-2/BP2, for example, this is a protein that has, I forget, several different RNA binding domains, has a very, very complex landscape of RNA specificity, and our model kind of struggles to accurately, capture its specificity, I think. and the clip data seem to be of very high quality, and so that's probably what's going on there. There could be other cases where the protein's binding in vivo is driven not so much by its intrinsic specificity, but by like partner proteins that
absolutely. Yeah, I can definitely see why the e-clip data will be better. I'm just sort of more confused the other way around. but maybe
so clip. I mean, when you see a clip peak, particularly one of these, ones that has a good IDR, is reproducible and so forth. There's protein there interacting with that RNA. But is, there are places where there's a lot of protein and places where there's a little protein, and there are vast differences in cross-linking efficiency along transcripts. And so I think that clip is, it's capturing the real stable binding sites, but then beyond that, there are some sites that the protein interacts with transiently while it's kind of searching for its binding site or whatever. And some of those just happen to have extremely high cross-linking efficiencies for whatever reason. They're U-rich or different, different technical reasons, and then you capture those as well. And then those, the protein does interact with there, but it doesn't interact, more than background really. And so it's not really predictive of regulation in those cases. That's our interpretation. Can't prove that, but we have various evidence that's kind of
that makes sense. So then it's a really nice way to see what are the bad places for clip and the good places in terms of cross-linking. And
this kind of relates to the observation I mentioned before, where you take clip peaks that have a high affinity motif in them and clip peaks that don't, and the ones with a high affinity motif are more predictive of regulation. And so we think that's because it's sort of a filter on the quality of but there are certainly going to be proteins where either the E. coli expressed version of protein doesn't behave like the endogenously folded version, or again,
right. That's
where the in vitro just doesn't really predict what it's doing. What it's doing.
Thanks. Really appreciate it.
Thanks for that question, Sylvia. Oh, okay, so I'll just wrap up here. So I told you about how large scale in vitro and in vivo data for human RBPs can enable construction modeling maps. For predicting RNA splicing activity, you could also do a similar thing for collision modulation or other activities. And the quantitative descriptions of affinity using these PSAMs can be useful for predicting binding locations along transcripts, and is surprisingly good at identifying likely regulatory targets of RBPs. And we hope that these will be both the sort of the data and these kind of modeling approaches will be useful for efforts to kind of sort of therapeutic efforts to perturb RBP binding pathways. And I just want to stop and thank people that did this work. So our RBNS data was generated by some really wonderful technicians, including Keegan, who's now in grad school at UPenn. Dan Dominguez, who's now on the faculty at UNC, was kind of the one who sort of enabled us to make this assay work at high throughput. And Marvin Jens, who's now back in Berlin, is the one who developed this RBP AMP method, mostly single-handedly, but with some input from Ralph Bunchu as well. And thank you for your attention. Happy to take questions.
Fantastic. Thank you very much, Chris. Much appreciated. We have a question in the chat, so maybe I will ask that one first, and then
sure,
you can have their questions in. The question from Sophia Adamia, she asks, can we use this approach in cells derived from patient bone marrow? How sensitive is this method?
I mean, yeah, I mean, I guess we could compare to clip data from bone marrow or something like that. I mean, our approach is purely in vitro at this point, purely using recombinant proteins in solution. But the how well it corresponds to and then we've compared to clip data, but the clip data is from cultured cells here in this case, which has been generated, sort of high throughput. But how that relates to then clip data from actual tissues and primary cells and so forth is a whole other question. The Yo lab, is starting to go in that direction, and that'd be very interesting to sort of really look at how it predicts, the true binding in an organism. Yeah.
Is anyone has done that in patient samples, like a fresh samples, even healthy donor bone marrows, lymphocytes, for example?
Yeah, I'm not sure about bone marrow per se, but certainly the Darnell lab has done work in human tissues. There's been quite a lot of work with mouse tissues from a variety of labs. Often you can use thin slices of muscle or brain or things like that. The cross-linking is tricky. It's not, UV cross-linking is just not a very efficient. It's just not very efficient. And so there are some tricks like Tom Tuchel's trick of using 4-thiol-U to improve the cross-linking efficiency, but then you have to culture things for a little bit to get that in there, and so that's no longer strictly in vivo anymore. So this is something where we need better methods. There are methods like this TRIBE method, which is a method with a recombinant. It's not particularly good for human, but for mouse, where you can engineer the mouse to have an editing enzyme fused, an RNA editing enzyme fused to your RBP of interest, and then it'll just edit in that neighborhood, and then you can pull out those sequences later. No cross-linking required. And that's a nice approach, that gets around some limitations. But yeah, it's tricky. These are tricky assays to do, and with patient material.
Okay, cool. And on the topic of tricky, Chinji Zhao asks, Zhu asks, can we adapt this approach to study double-stranded RNA and, for example, microRNA duplex binding?
Yeah, modified nucleotide. Yeah, if you can make the modified nucleotides in vitro and then read them out, or have barcodes to tell you which is modified or not, or have some way of kind of interrogating that, absolutely that this could be done. I do have some. The lab down the hall, Jean-Wei Li, is collaborating with Wendy Gilbert at Yale, who some of you may know. And her lab is doing sort of high throughput investigation of RNA modifications, pseudo-uridylation, and others. And they've actually done essentially RNA binding-seq with with modified RNAs in some cases, and it can be done. It's trickier, and we're not. Our goal is sort of to kind of capture the typical behavior rather than the special target that has the modification. But it certainly could be complicating the in vivo analysis for sure. Yeah, and then there's a question about. Oh, go ahead.
Yeah, so I was going to say along those lines, Fabrice asks, do you have RNA binding proteins that could be binding modified nucleotides, and could that explain the fuzzy profile of some of your motifs?
Yeah, yeah, we haven't investigated modified nucleotides in our assay yet. So it's that we are open to well, first of all, we're open to sharing our our protocol with anyone who wants to do this assay. It's not a super hard assay, and we're also open to collaboration. If you have ways of manipulating these modified nucleotides and want to work with us on that, we'd be very, very interested in that.
Yeah, I guess a place to start would just be to ask if some of the metal 3, metal 14, the ones that are known to bind modified nucleotides. Do you see, a fuzzy profile? Anyway,
yeah, we haven't looked at those proteins yet. Yeah.
Right. And then last question from Svetlana Lebedeva. She asks, could you in the end resolve the poly-U binding proteins? Could RBP-AMP predict which of them would outcompete another if they bind the same site?
Yeah. So that's a great question. So first of all, in our new RBP-AMP approach, we do allow multiple motifs, and in some cases, you'll find that that one protein really likes to bind longer stretches of U, the other shorter, or one allows interruption with an A in the middle and the other doesn't, or there are these subtle differences. And one interesting comment about this is that when we look at the CLIP data for different proteins that all bind poly-U in vitro, what we find is that we almost always get poly-U enriched in the CLIP peaks for each of these proteins, as you might expect. But the CLIP peaks, if we correlate the locations of the CLIP peaks between these different poly-U binders, they correlate poorly, okay? So what it appears to be is that they all bind poly-U, but they're able to bind distinct subsets of all the poly-Us that are out there through, it appears through sort of subtle differences in their binding affinity. And we can sort that out to some extent. Another question that we're interested in is sort of the evolutionary question of like, why, if you're evolving a new RBP, why would you evolve yet another, poly-U binding protein rather than going after some new specificity which doesn't currently exist in the space of RBPs? And we have some ideas on that, but there's none that are sort of ready for prime time. But that's sort of something we're thinking about. We think if we can understand the evolution, it'll help us kind of understand why it's set up this way.
All right, thank you, Chris. I think we'll end it there. So thanks to Chris, great talk. Much appreciated. And thanks to all of you for attending, your great questions. We had about 170 people on the call, so this was really great. So we'll see everybody at a future event, maybe even next week. So thank you very much, Chris. Much appreciated.
Great. No, thanks for all the great questions. Yeah.
All the best. Bye, everybody.